3 students with question, although that didn't really work last week, but we have 3 students with questions. So I don't know how to say that. Yun Wen and Teng Hui Xuan and Nguyen Quang Hui. Okay, so that's a Vietnamese name, I can tell. Okay, so there we go. I'm just going to put the names inside because I'm sure my pronunciation will not go far. There we go. Yes. Well, that's the
Right.
3 students who promised to ask you a
Right.
question today. Okay, but I don't know.
Yeah,
Maybe
amazing.
they yeah. I kind of I tried to contact them last week. No one really replied. I think they were kind of shy. Maybe there was a new
Mm.
topic for them. So, but I think
Yeah,
it's
absolutely.
it's a really interesting topic and it's going definitely in the right direction. And in regard to architecture or even other topics as well. I'm quite certain
Yeah.
that everyone can make something out of it. So
Yeah, absolutely. Yeah. Yeah, I just want to preface it by saying, yeah, thank you again for the invitation, Nicholas. And it really means a lot, and being able to disseminate my work and research is an absolute pleasure. Yeah, and then the word architecture, I kind of mean it fairly loosely in terms of scale. So we'll kind of go over what by design means in architecture and with the AI crossover. And yeah, I think the scale will be a nice, interesting segue to think about as well. But more to come in a minute. Yeah.
okay, thank you.
Okay, so let's dive in. So yeah, and also if you have any questions at the end, in addition to the 3 students that Nicholas mentioned, happy to answer these questions. Feel free to ask, jump in, do not feel shy, and yeah, we can have a good discussion.
Yes, please.
Okay, yes. Right. See. Yeah, okay. So today's topic, so going on from last week's topic, which was biodesign and biomaterials in general, where we talked about the fundamentals of biology and how living systems and materials can be used as a design material. And we briefly touched upon real life case studies and the limitations that emerge from the fragility of living systems and unpredictability and the temporality of these organic systems that would change the way we design with living things. So, very fundamental, very conventional, traditional form of what we call biodesign. And I deliberately made this transition last week to start thinking about digital interventions of by design. It's very well using organic forms and living materials to design with things. But if we layer in another extra technology, this time in the form of digital technology, how would that workflow change even further? And what kind of ethical and sociocultural implications that we could think about as a result? So this really comes in really nicely in terms of timing of the emergence and the rise of AI in many aspects of our lives. And design, academia, and research is not different. It's been pervading us for some time. And with the given acceleration of this technology and availability, my guess is that this will be a huge paradigm shift that's going to happen fairly quickly. They talk about doubling of these capabilities every 4 months, which is an incredibly fast pace. So I suppose we have to get used to it, prepare for it, and ask ourselves, take a pause and think, what does it all this mean? What's this artificial intelligence? How that can shape the way we work and how we research. Transition. Again, I don't want to go here again, but very quickly, if you haven't listened to my talk last week, this is my background. I come from a mixture of biology and design interaction background. For example, I have a master's in design interaction from Royal College of Art, and I also did a PhD at Queen Mary University looking at biodigital systems. And my core focus areas at the moment are working with living systems, making invisible biological processes understandable and accessible. And this is where my latter part of the talk will be, to really think about tinkering with molecules, tinkering with proteins that are not very visible in everyday life. And then, of course, during my master's, I was engaged in speculative design, um, explorations, and we can talk about speculative features that are grounded in real practice if we find some time. But essentially, these are my core experiences, and hopefully I can give you some value today in terms of how AI can actually impact the way we design with living systems. So, guys, when we talk about AI, for the most of you, or the mainstream general public to a certain extent, they think of generative AI and chatbots and LLMs like ChatGPT. Mid-Journey is a very old now example of how AI could be used. So this is one of the early explorations of how I could use things like Mid-Journey and other types of image generation softwares to think about how biodesign could be generated using these kind of tools. So these are very speculative prompts I used, thinking about how fungal networks, microbial protagonists, these are the microbial and bacterial protagonists we mentioned last week, of how this could be fabricated. And we kind of played around with this idea of engineering and fabricating living things to suit our needs or agendas. So this is a really whimsical, playful example of how AI could be used on a very low level. So this is a very fast generation, incredibly accessible tool that we can just use to think about what we could design with nature and what we could engineer from molecules like DNA and proteins. So this was a really interesting starting point of thinking about, okay, if we can actually generate these fictional and speculative artifacts through AI, could we fundamentally actually use AI to do real biology? These are complete simulations, of course, trained on past imaging data that are generated from real organisms. But if we set that aside and thinking about, can we actually change the way we fabricate nature and thinking about sustainable materials, can you actually use AI to produce something that's completely artificially generated, that's never seen in nature before? And this is an interesting question to ask ourselves as a designer in moving forward with this technology. So as I said last week, biodesign is actually about tampering and tinkering with living materials. There are also interactive elements that allows the designers to interact with living systems. And the third layer of the AI acceleration expands access and creative imagination, as I mentioned before. So I think the time is right now is to think about how AI matters and is becoming very relevant in biodesign. Certainly in the area of genetic engineering, certainly in the area of biotechnology. So one of these hard sciences and engineering, AI really matters at the moment. There are many discoveries, advances, and introductions of new tools and technologies within this space that gets scientists and engineers really excited. One of the main breakthroughs, I would say, within the last 10 years is this idea of having a predicting AI tool that allows protein structures to be predicted with incredibly high accuracy. I will talk about this specific tool in more detail. It's called AlphaFold. which has been developed by Google DeepMind. And there are other tools like generative AI tools like protein diffusion models that are able to generate new molecules and new proteins completely artificially. So these are generated by algorithms that's never seen in nature. And then another areas of excitement within the hard sciences at the moment is this idea of autonomous labs. Someone mentioned this idea of self-driving laboratory. When I heard that phrase, I immediately thought of a car with a lab inside, but this is autonomous driving, as in the self-driven laboratory that allows the experimentation to go ahead, assess the data, analyze it, and make sense of these analysis in a completely autonomous way. So robotics um, and yeah, automation is a very big thing in biology right now, especially in terms of automating and self-driving these scientific endeavors, which is a very profound move at the moment. And last but not least, a lot of cost is involved in running labs and engineering spaces. What AI can do also, like the one we showed you earlier, is the biological simulation and modeling predictively of how these biological forms could actually be generated. So let's talk about AlphaFold. So I would say within biology and also widely in biodesign, AlphaFold is probably the most important invention. Some say this is probably the most useful application of AI at the moment, which is to predict how living molecules like proteins could be shaped. This might sound very arbitrary, this might sound very cryptic and conceptual, but everything that we see that's living or organic, they are made out of proteins, and proteins, the way they fold and they shape, it determines how they function in the real world. So think of it as a living origami that produces a 3D structure that can produce a certain functionality. So AlphaFold is driven by uh, machine learning algorithms to generate predictions of how a protein sequence would fold. Going back to the basics, the DNA sequence would determine the protein sequence, which then would allow the proteins to shape in a 3D fashion. And what AlphaFold does is simply predicts with a very high accuracy of how these things would fold. And this is one of the areas where the notion of invisible things being made visible through AI is an example here. And it has also a vast public database that this algorithm works on. So if you think about decades and decades of biological research and ton of data has been injected into the public databases, like PDB, for example, what AlphaFold has done is they've learned to see how the proteins fold by learning from these huge swathes of protein sequences that have been injected by scientists over the many decades. So this is one of the pinnacle tools that allow the vast amount of data that's available to predict and allow us to fabricate and imagine how these proteins would be folded. And one of the key proponents or the pioneers of AlphaFold is Demis Hassabis, who is a very famous person. I'm sure most of you have heard of this person. He is a head of the isomorphic labs, which have not only invented AlphaFold, but they're also running new projects that are more scalable, more autonomous, and they're actually coming up with newer versions or new models of AlphaFold at the moment. So it's very exciting times for Demis Hassabis and Isomorphic Labs. And there's a lot to learn from biodesigners in terms of how we can actually leverage AI for biodesign. So AlphaFold would be very disappointing if we didn't give you some case studies of how AlphaFold has been used in real life. So back in 2022, it's already 4 years ago, AlphaFold was used to predict a protein for a novel drug target for Plasmodium falciparum. So in a nutshell, what they did was they used AlphaFold to predict a drug target to attack malaria parasites, which then would effectively kill the parasite, uh, kind of render it useless so that the malaria could be treated in this way. And there's a lot of headlines generated. AlphaFold is helping to defeat malaria. There's a lot of hype and noise, of course, to really filter out these kind of mass media headlines. But essentially, it kind of shows or hints at how AlphaFold could be used to design new drugs, to tackle modern diseases, and traditional forms of medicine is being revamped through tools like this. There are other models, as I said, AlphaFold is one thing, but protein diffusion model is, I would say, more interesting version. So if you think about generative AI, this is it for biology. So these models create novel structures from scratch, rather than just predicting the shape of existing ones. That's been illustrated by the AlphaFold example. And one analogy I would put here is a thing called the molecular mid-journey. So similar to the image diffusion models like Midjourney, but on a more of a applied through molecular geometries and biochemistry. What's interesting about the RF diffusion model is that it goes beyond the natural forms. It's capable of inventing protein shapes that are completely alien to us, that are functions that are not found in nature, and opening new design possibilities as a result. And again, it has diverse applications, which have been making the headlines in recent years. So one of the examples of how RF diffusion, the AI-generated models, could be used is in the field of enzymes. Again, it sounds very engineering focused, but this also matters in biodesign because enzymes are critical for generating new textures and new physical properties for a biomaterial that you're working with. So if your mycelium or your bacteriocellulose material needs to be changed in terms of their physical property, whether it's rigidity, flexibility, enzymes could be engineered using AI to really attack these surfaces and to modify the biomaterial in question. And again, back in 2022, it seems like this year was a golden year of discoveries using bio AI to generate enzymes that are used for industrial purposes. I'm not sure whether how much detail they go into what type of applications there are, but certainly there are industry focused applications which would have no doubt a great economic impact for the society. Yeah, and last but not least, the autonomous labs and robotics, as I mentioned earlier. One of the recent news I heard just last week is that Ginkgo Bioworks, so this is it's a huge synthetic biology company based in California, I believe, in the US. So they raised a lot of venture money to create autonomous labs. Um, yeah, and it's really interesting to think about the motto they used for their promotional materials. The concept is kill the bench. So this notion of traditional biologists or traditional engineers working on a bench, sitting as a person, pipetting, calibrating, and measuring things on the bench, that's no longer the gold standard. Their proposal is to have AI-guided experiments with robotic arms doing most of the grunt and physical works, which reduces human intervention and error, of course. And then this is a closed loop design cycle. It has this design, test, iterate cycle, which can be automated, obviously. And again, it really crushes this idea of scientific validation. Who can we trust? What are the trustworthy workflows? What are the data that's trustworthy? How can you trust it if AI is generated? That's a whole can of worms that we can talk about in terms of the ethical aspects. But this is what's happening. Everything is being um, collapsing in terms of hierarchy. Everything is being automated. There's less human intervention. So this is a very exciting, but also a critical time for everybody to think about what it means for us as humans and what value we can actually provide in the age of AI. Yeah, and then talking about this idea of collaboration. If you remember last week's talk, we talked about collaborating with the non-human, collaborating with the microbial. So thinking about strategies to collaborate with microscopic organisms and the strategies that we can use to effectively for us to notice microbes, to collaborate with them. But now the paradigm has shifted again, where the AI or the machine learning side of things could be harnessed as a collaborator to engage in automated experimentations and different possibilities that we can do with biology. Yeah, so this is one of the promotional materials. Um, yeah, I don't think this is Ginkgo Biolabs, but it's the other lab based in the US. And yeah, you can see there's a lot of robotic arms, programming, and software involved. And this is, I think, also creating new jobs um, in the marketplace. There's a huge scare amongst us with the AI taking people's jobs, but I think it will create new jobs and processes and workflows, which I'm happy to share as well. Yeah, and this slide is really apt. I think this is a really nice one because it allows us to think about biology not as materials, but as data. So this is one of the ways of thinking that we could adopt. Uh, by design, by tradition, it's all about materiality and materials. But if you think about data also as material, it might be a better way or more productive way, meaningful way to think about designing with biology using AI. Because AI doesn't really care about materials. It doesn't see the materials. It cannot feel the material. All it sees is data, tons of data, things like DNA sequencing data, biochemical composition data, physical properties data. Let's not forget tons of microscopic images that can be generated, and also time-lapse videos to capture the temporalities of slow organic forms. These are all data. So if you can think about biology as data, I think it might be a better way to think about collaborating with AI. And through some demonstrations later on in the talk today, I can kind of illustrate how the data can be made meaningful for these algorithmic tools. Yeah, so just to sum it up, why AI works so well in biology, especially things like AlphaFold and RF diffusion, because they absolutely love patterns, right? They follow patterns. The biological systems follow patterns that can be recognized. Humans may not be able to do that because of the limited senses that we have and cognition, but artificial intelligence, they may be able to churn and crunch a lot of data to create and recognize patterns that are invisible to us. And we can also give them constraints. Obviously, biological materials do have constraints, whether it's physical and chemical, and AI can be trained on these constraints to generate certain models. Biology is also, if you think about it in terms of um, dogma of biology, going right from the beginning of the DNA base pairs to proteins, cells, tissues, organs, and organisms and colonies. So these are all very hierarchical. And what better way for AI to get their hands on than rather than like a folder structure or these filing systems of biology. If you can translate these filing systems in a meaningful way that AI can understand, then we can actually allow AI to almost like an agentic system to control these filing systems, to sort out and recognize patterns, and to generate new models. And there are other aspects, things like the high dimensionality. There are complex interactions across different scales. So if you think about molecular interactions, which is completely um, yes, some people might find it so abstract and very conceptual because you cannot see, even with the aid of a microscope, of what's going on. So you have to rely on inferences of chemical changes or number changes. But yet AI can actually process these things in a much faster way. Also, biological noise. We mentioned last week the messiness of biology. We cannot make sense of them, but maybe AI can. There are a few studies out there that study these random mutations that the cells produce, but AI can actually make sense of how and why these seemingly random mutations occur. So it's a really exciting field at the moment. If you look at papers like Nature or Science, so very highly esteemed journals, a lot of scientific papers are emerging at the moment of how the random mutations can actually mean something for medicine and for the sustainable energy designs. Yeah, and I very briefly mentioned the central dogma of biology. I won't go too much into detail of how the biological things works, but it shows the really nice hierarchy of how biological things we understand and how AI can actually intervene in all of these three layers as well. And again, drawing an analogy of the design of the form follows function, it's kind of a similar way, but on a molecular and organic context, which we can think about as well. Yeah, this is you probably don't need this slide at the moment. One thing that's worthy of note is there's another protein folding tool called ESM Fold, which has been developed by a company, Meta, of all people. So Meta is obviously famous for Facebook and Instagram. But it's really funny, right? Because to question why Meta has been developing biological tools like ESM4. So it kind of goes to question their intentions and, yeah, the ethical implications that we can also think about. Yeah, another concept that I like to introduce is this idea of the AI as a search engine. You can use AI to explore genetic possibilities. I mentioned the vast databases and networks of biological data. And you can train AI to search for clues or search for new ideas of how by design could be carried out. So if you're looking for, let's say, an abstract material with a certain functionality, you can use AI to search these databases to actually mine and pick up different structures and different forms that you can generate using these databases. So it's a really handy tool, not as just an engineer, but as a designer thinking about the end goal or the outcome of how biological form could be shaped. And again, it can be optimized using training to pick out the best performing sequences within these databases as well. Yeah, just a little bit more on AI being used as a simulation, simulating collaborator, binding dynamics, folding processes, system assembly. So these are the vocabularies that are used by everyday biologists, but we can actually put them into the tangible way to visualize and surface, or let's say notice these molecular interactions using AI. Obviously, this is not completely new areas of implementation, But what AI does is it makes it more accessible. For example, Chimera X and PyMol, for example, they are really classic examples of molecular visualization tools that are accessible that we can use AI to speed up. Yeah, so it's very well talking in terms of how engineers could benefit from this. I would argue that bio designers could also benefit from these technological advances. Firstly, as a practicing bio designer in the past, who struggled a lot with finding laboratory spaces, contacting and networking with professional scientists to access their laboratories. It was a real pain. But now, with this AI tools that are available, it sounds like a cliche, but this is actually true. A lot of biological explorations are now happening outside of the lab. As we mentioned, not least with the autonomous labs with robotic arms. IoT is a really big implementation. These automatic mechanisms that are networked, data can be uploaded onto the cloud. So everything has to be, uh, it doesn't have to be within a physical space. So a lot of people are moving away from the labs, but also visualizing, predicting, and analyzing data that can also be done outside of the lab, obviously, using these other four tools and RF diffusion. So, as a bio designer, certainly there's not much better time than to be uh, harnessing biology and design at the same time because of AI and machine learning at the moment. I know it sounds a little bit utopian. There's a lot of ethical questions that we can think about, and I will cover these in great detail in a moment. But what I can say is that, yeah, designers no longer need to be fearful. They can have a go, can-do attitude. I really feel this way, that you can actually remove barriers of accessibility and do your projects. Um, and if you want to do a project based on this AlphaFold or other types of tools, I mean, I can also give you help after this talk also. Yeah, so AlphaFold 3, that's the latest model. I'm not going to go too much detail. RF diffusion, um, simulation. I'll skip those. Yeah, and now just to spell out some use cases of in biodesign of how these tools could be used. As I said about mycelium and bacteriocellulose, you can actually control how these textures actually um, form at the end of the cycle. So you can create novel surface properties inspired by biological structures. Thinking about in terms of interfaces, we can actually simulate interfaces that allow living system to interact with physical environments. Yeah, and thinking on in terms of genetics, you can think about programmable matter to fabricate your own DNA to produce a certain functionality. And again, I'll give you a concrete example of how you can do that. Okay, so this is the part that many people might feel uncomfortable or comfortable depending on where you're coming from, whether you're anti-AI or pro-AI. So in terms of risk and safety and the ethics that follow very closely, this is the part we can talk about the ethical side of AI. So biology reproduces, it self-replicates. AI also does the similar thing, but AI can also generalize and also find patterns. And these are very powerful in terms of scale. It can scale quickly, but at the same time, it can behave unpredictably, and they're hard to fully understand. So there's this whole idea of black boxing in biology. We actually don't know what the hell is going on with inside the cell, these wonderful chemical processes that happen and the mutations, as I said. But AI is no longer, it's not different, if you ask someone to explain how yeah, image generation works, everyday person might not know the full mechanism or the workflow that go behind it. So these are one of the examples of the risks that people should be aware of. And my point here is that because we are layering in the technology, we are actually compounding not just the benefits, but also the risks that are involved. Yeah, so just to spell out what I mean by this is that these technologies increase speed. They allow a very small window of reflection and examination of what we created. In the old days, without the use of autonomous robots and algorithm generated proteins, we have to wait until things produce because if you want to produce a biomaterial, you have to wait for it to grow. You have to culture it, you have to purify it. But now this can be collapsed using AI. So the speed is very it's a critical component to think about. Our understanding decreases. This idea of brain rot, with the idea of using ChatGPT to yeah solve anything and everything. That is also a similar problem in the biology field. If you can just ask AI to produce a molecule, do we actually understand what goes within the cells, or do we know what species we are working with? And then, as a result, the responsibility for designers is unclear. You know, who is in charge of who is accountable, who logged the process, who documented it, and ultimately, who is to blame for it. So the point is that the ethics becomes invisible unless it's baked in into the whole process. And we'll talk about how we can do that. Yeah, so I've listed five different entry points as designers for people to intervene within this biological workflow. I guess the main intervention point is right at the beginning of the biological process. So if you're, let's say, designing a sustainable textile, we have to frame the problem in a responsible way to give it guardrails of what the algorithm should not do, and yet the different responses that we should take should some things go wrong. So if a mutation that's very harmful happens, how should we intervene and stop the process? And so this leads nicely to the interface structure of how much we can interfere within the whole process itself. So could you create different softwares or interfaces, or let's say if you're working with a bioreactor, can we design something that would allow us to notice something that's going wrong, that's not visible to the human senses. And what are the default constraints and guardrails that we should put, as I said. And then just to reiterate on the idea of visibility and hiding of these risks and um, ethical factors. What are the ways we can actually bring this to the front? How can we allow people to talk about it? How can you raise awareness of these issues? And then safety perception is another big thing where designers can intervene. Um, there's a lot of talk within microbiology community of how they are perceived, whether these are harmful and pathogenic materials or they are very sustainable, gut-friendly, and regenerative materials. I think the designers have a lot to say in that of how outcomes and how communication is done to produce outcomes that are perceived in a safe way, not to cause alarm or panic. So these things are already happening in biodesign. People are designing for responsible and regenerative materials. But if AI is also implemented, that may need to shift or that may need to be modified accordingly. We haven't really seen a case like that at the moment, but it's something for us to think about. Yeah, and the invisible power layer I talked about. Yeah, and then reframing safety. not as something that's pathogenic per se. If AI is involved, it's maybe about the misalignment of values. So we are now getting into a nuanced idea of value systems. If AI, like code and biological code, DNA is involved, how can you actually bake in different values to create living systems? It doesn't have to be an enzyme that produces a lot of money. It doesn't have to be commercially very attractive uh, sequence of molecules. It can be something that's more friendly to the geographic environments or the local regions. And this is what I mean by reframing safety towards these values that are not very obvious to, let's say, capitalists and commercial agendas. Yeah, there's a lot of yeah. So this actually was like a workshop material that I developed. So there are different workflows you can think about as designers to find what are the potential ethical ramifications and how can we intervene as a result. And I'm not going to go into too much detail of how these things are done, but I can share this with you after the talk, and you can also run the exercises for yourself. So yeah, you can think about different scenarios of how engineers and other people involved could actually avoid blame when responsibility breaks, and how can you intervene in a responsible way? How can you reclaim responsibility? And how can designers translate this invisibility of power dynamics and yeah, the gatekeepers of access? So one of the questions I allowed the workshop participants to think about is to, yeah, these three fundamental questions. What should never be frictionless, meaning that what should never be automated? What certainly must stay visible to the human's senses? And where should designers say no? Where is that red line that people should say no and do not let that process happen? Yeah, and then I developed like a strength test tool. Again, this is a really handy tool for biodesigners to think about in the lab or outside of the lab to just run through these five different questions. Yeah, and you can also use this stress test before you carry out any type of project that you wish to do. If you're doing a graduation project or a biodesign collaboration project, I think this is a handy workflow to think about. You know, summarizing the project in one sentence, the AI's role, the biologist's role. So thinking about these two roles and the stakeholders, you know, who are the beneficiaries of your design, who are the people who might miss out in terms of accessibility, and then pick top two risks, one from AI and one from bio, and think about how to mitigate these risks in your workflow. Yeah, and then you can also run this exercise to the second half of your activity. So we will probably do like a role play. You come up with a bio design of your choice, and then you have to defend your design as if you're in a critical review session. So we would pick your design in terms of risks, like you pick one risk and identify it, and then you choose a challenger for you to defend your design in terms of ethical consequences, and you learn how to formulate your response, how you would use either a structured script or a workflow to generate your response that is defensible. Yeah, and it's a really nice exercise for people to think about if you want to start a project that involves either bio or AI or both. Yeah, so without going into too much demo side of things, I'll leave it here for now. But one of the things that I'm learning at the moment is that as much as we claim to know many things, and as much we think that AI knows a lot, I think there's a lot of learning points that people should be aware of, that we don't know everything, and we shouldn't rely on AI too much. We always have to slow down and put guardrails and be critical at every point of the workflow. And I think that this is the mindset that the designers, certainly the by designers of today, should adhere to. Um, so that's pretty much it for the first half, just to give you a general overview of how AI is impacting biology. And then the second half is probably be going to be spent on giving you like a short demo of these tools that I mentioned earlier, just to give you
Yes.
some taste of how these things could be applied in real life. We'll go into slightly more into detail of molecular design, this idea of architecting with molecules, and we can kind of nerd out on characters of proteins, characters of specific interactions that can happen within this molecular space. And again, these are all very accessible and learnable tools that you can use as well. So that's it for now. And if you have any questions, feel free to ask me.
Yeah, and then you can, of course, ask your question orally or leave in the chat as well. That's absolutely fine. Yeah, we all
Okay,
have access to the
wait
chat, I
one
believe. So,
second.
if
Okay,
there's kind of any question that you're thinking about during the break, feel
just
free
need
to type them
to
into the
exit
chat.
the thing because I think your
Do you have access to the
mic
chat?
is muted.
The chat is muted. Oh, they don't have access to the
I
chat?
think I can
All
hear you
right.
talking, but wait.
Ah, can you hear me? Can you hear me?
Oh yeah, you can. I can hear you now. Yeah,
Okay, okay. Now I was just saying that if they don't want to ask their question orally, they can also leave the question inside the chat. Yeah. I thought
yeah.
there was a problem with the chat, not my microphone, but okay.
All right.
That's fine enough. It's very good. Thank you very much. I really like your some of the ideas there, particularly like don't escape from the uncertainty. I think that's a really really good point. Yeah. And
Mhm.
you do you know have you came across this idea of cosmopolitics from Bruno
Customer
Latour?
politics.
So
No.
I mean, it is the idea that. Somehow we move from a traditional society to a modern society, and all of that remains within the confine of a very certain society. Modernity is
Mhm.
about certainty, and particularly in relation with nature. And now we
Mhm.
are moving into, well, for quite some time, we've already been moving into an area of uncertainty. But then again,
Mhm.
we have this choice of whether we want to choose an area of detachment, kind of thinking, like you were saying. I think AI somehow is a detachment from reality. The idea that
Mhm.
AI somehow supports this vision of the world where everything is dematerialized. AI can somehow say whatever it
Mhm.
wants without necessarily connecting with the consequences with what it says. Whereas
Mhm.
humans, well, not all humans, some humans with a standard level of ethics will start thinking, okay, but if we do that, what will happen?
Mhm.
It seems that people like Trump have totally kind of disconnected with this idea of consequences. So
Yeah.
this is really worrying at that like of level. You can see how somehow it kind of collapses towards the American population as well, and somehow it's like, well,
Yes.
let's do it. It's not a problem. But so
Yeah.
from this kind of detachment, so you would have some kind of form of relativism, which is to say
Mhm.
that everything's equal. Whatever the result from AI is the same as the result that a human being has been researching for a
Mm.
while. Everything becomes equal somehow.
Mhm.
And then
Yeah.
so Bruno Latour and Isabel Stengers, I'm sure you heard about Isabel Stengers, who work with the Nobel Prize in chemistry, Prigogine. It's been a
Mhm.
long time, but she was always promoting a slow version of science, for example. This is something
Mm.
you did say as well. I think I think if you haven't read her, I think you should be reading her because she's really fantastic.
Yeah.
But then, so this side of cosmopolitics is to say, okay, well, how do we face uncertainty? And then the idea, and I think from what you say here, it kind of confirms that the idea of moving towards detachment, more detachment, like AI is somehow kind of telling us to do, seems to be the wrong way. We, as humans, need to find a way to come back to a form of attachment, and that's what you were saying in the previous talk. Somehow,
Hmm.
that's quite interesting. And you
Yeah.
um, one moment you were comparing, the collaboration with the macrobio, yeah,
Hmm.
and then and that you were saying, I think everyone, I felt, oh yeah, that is true. There's
Hmm.
a second of correspondence, even though there's so much more than us, even though
Yeah.
somehow at this point we kind of manipulate them a little bit and don't think too much about the
Hmm.
ethic of it. Maybe they're not sentient, you were saying last week, but
Hmm.
maybe they're still alive, and that's maybe
Yeah.
enough someone to kind of realize that, okay, maybe we need to tread a little bit carefully there. And
Yeah.
the AI as a collaborator, as you were mentioning, I think that's another collaboration entirely. That thing
Hmm.
is neither sentient, neither alive.
Hmm.
It's more of like, like they were saying that's more like a parrot. It's, at the end of the day,
Yeah.
it's kind of a stochastic parrot. It's just
Yeah. Yeah.
kind of,
Having
it's
said
really
that, I
good at
think there is
repeating
a
and
new,
analyzing. Yeah.
yeah, yeah,
I think
yeah. I
there's
mean,
a,
the AI
go
sentience thing
ahead.
is
Yeah.
another huge can of worms, right? Because I think some people are actually talking about AI being sentient now, being conscious,
I
because
know, I know. But
but
I would be
yeah,
worried.
that's another whole
Yeah, but
argument.
again, I think to me this is a reaction to the uncertainty of things, and towards the uncertainty of things. And I think that's an interesting thing where somehow they want to find a way to get back to attachment.
Mhm.
So if again, this the cosmopolitical idea is actually okay. If it's detached, let's try
Yeah,
to bring it back to attachment. But that
yeah,
process of attachment, as Bruno Latour says, is not a
mhm,
double click procedure. It's not just like a
mhm.
click, click, and boom, it works.
Absolutely, yeah.
It's a
Yeah.
process of composition. It takes
Mhm,
time. It takes time to kind of say, well, okay, AI. I'm not pro or against AI. I really don't
mhm.
care here. I'm trying to be critical. I'm trying to say, okay, there's some good stuff that will help
Mhm.
us. And
Yeah.
definitely in some situation, we need some help. And like you were malaria or maybe Ebola, maybe I guess maybe it can be used
Oh yeah.
somehow to help us with the current situation happening in the States at the moment. All of these
Yeah.
things, I think I think great. Yeah, that's not a problem, obviously.
Mhm, mhm. It's just the
Mesa, you
speed of things, isn't it? It's just
yes,
so
yes, yes,
so quick, and they're
yes,
saying that they're generating
yeah,
so much
yes.
publications and data at the moment. We
I
just
know,
simply don't know what to do with them,
I
let
know.
alone
Yeah.
understand them, you know?
Yeah, I know, and the whole peer reviewing system is really not following up with this kind of
Yeah.
thinking. That now you
Mhm.
actually see AI peer reviewing system. I don't know if you try those kind of systems, just like,
I
yeah,
did,
but
yeah.
no. Because every time your idea kind of, because there's a problem of normalization with AI as well. This tendency with
Yeah.
AI wanting to kind of format thing in a certain way. And then if you, as designer, we love to kind of, as speculative designer as you are, we sometimes we like to kind of, take a bit of
Mhm.
a lateral tangent. And AI
Yeah.
really doesn't like that at all. Yeah,
Yeah,
that's
indeed.
not the way that it thinks. So it hasn't
Yeah.
been prepared for that. Yeah.
Yeah, yeah. It's troubling times, because
Yeah,
how
no,
do
no.
we,
Yeah,
as an academic like yourself,
yeah,
how do you reconcile that? This
no.
idea of credibility and validation. Where does that come from it now?
Yes.
If
No, I
it's not a
know.
publication, then what? And if you're working with something that's
Yeah.
seemingly more intelligent, academics is all about intelligence, and
Mm.
if that's being compromised in some way, that's a very troubling time.
Yeah, yeah. I mean, there are processes. I don't know if you know the research through design conferences. I mean, it hasn't happened for a
Mm,
while, but it happened. One of them happened in Delft. I don't know. You were in Delft
mm.
2019. Uh, the RSD conference as well seem
Yeah,
to kind of
it's happening in Edinburgh, right? Yeah.
find ways of bring the human back into the peer reviewing,
Mm,
and kind of engaging with discussions.
yeah.
I was a reviewer at the DIS conference in Singapore, and
Mm.
there was the
All
same
right.
thing where at some
Yeah.
point where we're kind of put into a forum and we have to kind of, it's not just we can say whatever. At the end of the day, there is a
Mm,
discussion happening, and I think
yeah.
that's quite good because if
Indeed.
any of us, had a stick, in someone else's
Mhm.
paper, we could tell
Yeah.
really quickly.
Yeah.
Any of us brought something with AI, we could tell quite easily
Yeah.
as well. I think still we
Indeed.
can
Yeah.
tell. I think there's
Yeah, I think there's a lot of
a
I think with
rhythm.
that conference, I think a lot
Yeah,
of people were rejected because they used
yeah.
fake citations. We had
Oh,
to, I mean,
oh,
I was not a
oh,
reviewer, but my partner reviewed some and they had to desk reject like
oh.
maybe a dozen of papers because they seem to be generated by AI. So
Yeah, no,
yeah.
okay, wow, yeah, wow, yeah. I had three papers. I rejected them because none of them made any sense whatsoever. I don't know if that's an AI problem, if it's a human problem. Okay, fair enough.
Yeah. Indeed.
Okay, great. Let's have a little quick break. Yeah,
Yeah.
maybe we can meet again in 10 minutes. Is that okay? Yeah,
Yeah. Yeah. Sounds good.
7:40. All right, let's have a
Yeah.
break off
Great.
the screen. Maybe that's my
All right.
father's
Yes.
thinking here. It's just kind of thinking, let's
Okay.
get off our screen. Too much screen time. All right, see you
Yeah.
soon.
I'll get some coffee. Yeah.
Yeah, good idea. Not me. See
Yeah.
you.
See you. All right.
Hello.
Yeah.
Oh, just one question. Did you know Joseph Popper? Were you in the
Yeah,
same?
I studied with him at the RCA. Yeah.
Oh, okay, okay. I met him in Milano in what was that? In June, last
All
June.
right. Okay.
This was a year ago, but yeah.
Oh, right. I think he's in Switzerland,
Uh, he
working
was
in
teaching in
Switzerland.
Vienna. He was doing his
Ah,
postdoc in
Vienna.
Vienna.
That's the one.
Yeah,
He did his
but
PhD in Switzerland.
yeah,
Yeah.
yeah, that's correct. Yeah, yeah, that's correct. That's
Uh,
correct. Yeah, yeah. Oh, good. Yeah. Oh, so you both are connected. Okay.
yeah. I mean, I haven't spoken to him in a while, but yeah, I think he's doing okay.
Yeah, no, I'm trying to get a bit of money here to organize a conference about. I've
Mm.
been researching objects in cinema and then the relation between cinema and design, and obviously it
Oh,
would be
yeah.
great on
Mm.
that topic, and then we'd be talking about that. But it was really fun because I mean, we didn't know. We both signed the paper to a certain kind of theme track in a conference
Mm.
at Politecnico de Milano on the STS
All right.
conference. And then
Okay.
we thought, oh, we're on the same theme track.
Oh, I see.
So there was
Yeah,
four people talking, and then we were, I mean, we were there. It's so
I
fascinating.
see. Yeah. No, I think he'd be great to have actually, to as a
Yeah.
speaker.
No, no, of course, of course.
Yeah.
I'm just trying. It will. I'm absolutely. It would be great to have his workshop as well. He had a really good workshop.
Hmm.
Yeah. So what
Yeah.
sort of workshop are you doing right now? Is it just you were talking about this workshop? Is it a very
Yeah.
more theoretical workshop about ethics and
Yeah.
design
I
and
mean,
biology, or is there something of practice as well?
yeah. I mean, it's really tinkering with these AI tools on a molecular
Okay,
level.
I see.
I'll give you some quick demos of what we've been doing with
Ah,
the
okay,
workshop
yeah.
people, but yeah, it's something that I can offer later down also down the line if you want.
Yeah, yeah, of
Yeah,
course.
but it's one of the kind of side projects that I'm doing at the moment. Yeah.
Oh, okay, okay. I
Yeah.
definitely, if you have like some sort of brochure or any kind of anything or kind of example video that you
Mhm.
have made, I can try to kind of
Yeah.
see if there will be a public for that,
Yeah.
or
Yeah.
if we could kind of make it maybe more specific to the world of architecture, for example, in terms of
Yeah.
we're talking about mycelium or talking about
Mm.
I really don't know in terms of AI based biology if there's kind of an element that could help. Maybe it's something
Yeah.
to research. That could be a starting
Yeah.
point
Indeed. Indeed.
to something. But
Yes.
yeah, if you come
I
up
can
with some
certainly
ideas, I
send more
mean, you can always
details.
write. Second,
Yeah. I can certainly give you more details of yeah
yeah,
what we can do. Yeah.
great,
Great.
thank you. All right, so if you, I mean, so
Yeah.
you say you want to kind of show us a bit of some example
Hmm.
of the tools that you've been talking about. I mean, that would be very welcome to have a little bit of
Yeah.
practice and to see how things are being done. I think it's
Yeah. No
awesome to
problem.
that'd be great.
Yeah. Okay. Yeah. So this part of the talk is really thinking about tools and demonstrations. I want to also start by giving you some examples of petitions and initiatives. So there's a thing called responsible AI in by design. So there's a huge community that's growing at the moment,
Nice.
concerned about this, the very fact that AI can actually steer how biology is done and how it can be biofabricated. And there's like a committing form that you can also sign with your names. So essentially, committing yourself to be more responsible or responsibly aware of the potential consequences of AI in biodesign, and you're happy to also add to the list, add yourself to the list as very interesting in terms of the names that are being presented, coming from not just within bio design and science, but all walks of life and workplaces. So interesting to look at. And then last month, I was in engaged in a panel conference for Chi conference. So we gathered a lot of interest, and we invited some panelists across different areas of design to really debate and think about what are the consequences of integrating AI in biodesign. So we did a whole debate, which was really successful.
Nice.
Here is a reference paper you can also check out. I can also share that in the chat. Yeah, so do check it out. We will publish like a report from the panel, so you'll be able to track and yeah, read more about what was discussed and the next steps that we proposed. And then last but not least, biodesign in the age of artificial intelligence. So this came out quite a while ago, for 3 years ago. But it's an interesting reference for you to read about if you're interested in yeah, reading more about this field. It is quite expensive, but I believe yeah, I think the institutions can cover the yeah, maybe it's in the library already.
Yes.
Let's see. Okay. And then don't forget to sign up for Biodesign Academy newsletter if you haven't done so. If you want to continue the conversation. So I try to post every week within this topic, especially in terms of biosafety and yeah, designing with molecules responsibly. So it can be a little bit nerdy and conceptual, but if you're really into molecular design, then I think it's the sweet spot for you guys. So yeah, in any case, sent. Okay, it's a bit of a lag here, but let's see if you can get it up to the presentation. Next one. It's stuck, but wait.
Take your time, it's okay.
Yeah. Oh yeah, so
Okay.
yeah, so basically I will give you a quick demonstrations of some of the tools that you can use to predict protein structures. Again, if you are into this kind of thing, you'll probably love this one. If not, don't worry too much. So ESMFold is open source tool developed by Meta, and what it does is it allows you to predict structures using AI. So this is what we call a standard format for displaying a protein sequence. So these are sequenced molecules that's represented by these characters. And there are 20 different characters that can be strung together, like this one, for example. So if you have a desktop at the moment, you can also try yourself, but if not, don't worry too much. You can also check it out in your own time. Yeah, so you fold the protein and then you can generate different shapes. So in the workshop, we go through how the shapes are generated and what the shapes translate in terms of its properties. And if you are specialized in, let's say, mycelium or bacterial cellulose, which is also being poised as one of the emerging architectural materials, the whole point about this workshop is that yeah, you can fabricate and predict the different properties of these biomaterials, and I can give you like a workflow that you can also borrow as well. Just to reiterate the point about the diversity of shapes and textures and conformations of proteins. If you think about the complexity of life, there are literally probably millions, probably billions of different conformations of how these proteins could be folded. And there are different types of proteins with varying sizes. This one is much bigger than the previous one you just saw. As you can see, there's a lot more going on here with different shapes and conformations. We also talk about the confidence levels. So now we're getting quite technical. So the different colored regions of the molecule is basically saying how confident this tool is predicting a shape to be. In a nutshell, proteins are usually in three different shapes. So one common shape is this kind of well, it's almost like a fusilli shape, like a DNA helix. It's like a spiral shape. So this is one of the most common shapes that protein folds into, and it has a certain functionality. Second shape it can also fold into is these arrows. They look quite alien to conventional graphic designers and everyday, layperson, but these are what we call beta sheets. So molecules have a certain way of folding into these sheet-like structures rather than being coiled. You can also have very loose rope-like structures like these parts here, and they provide a lot of flexibility and elasticity to the material itself. So from a designer's point of view, this is very interesting because if you are given an unknown sequence, whether it's from nature or whether it's artificially generated, you can use these types of softwares to predict how the shape's going to be once it's folded. One thing I forgot to mention is that these things would be inside a living cell. So these will be folding inside, let's say, a bacterial cell that would produce this protein, which then would become a matter or a material, the actual physical component that you can use. Um, yeah, so this is an interesting tool to think about. And then the second one, I mean, there are tons of tools, but these are two most commonly used. So this is what we call AlphaFold. And you can actually run it using your Google account. So if you just run this one, let's say, so it will
Yeah.
generate a similar sequence prediction as the ESM fold I showed you earlier. But this is more complicated in terms of the different parameters you can adjust. You can calibrate the type of the type of training that you can actually implement. So it can be very technical, but it just give you a flavor of how people are using AlphaFold at the moment to design proteins. A lot of startup companies are using this as a wrapper. So a lot of digital products these days are ChatGPT wrappers. And in a similar way, AlphaFold is no different. They use AlphaFold as a wrapper to design, let's say, an array of drug targets or array of um, uh, energy generating molecules or a function X. So you can actually create a lot of businesses and enterprises using this AlphaFold as a wrapper. But let's see if the protein is being generated. And you should be able to get protein sequence. So this is just the analysis. I think the running of the image generation is a little bit slow, but we can come back to that later. And then we'll talk about the reading of the structures. As I said, again, I don't want you to use this as a workshop session, but this is just to give you an idea of the vocabularies and the levels of technical details that engineers are going through at the moment. So helices, I mentioned, sheets and loops. So in many ways, these are very architectural vocabularies that can be scaled up to an architectural level. Um, And then you can be color coding your regions of your biomolecule that you designed using confidence level indicators of blue, yellow, and red. So in general, blue is a high confidence that you can actually trust this prediction. And if it's yellow or red, the AI is not quite certain. So it's very hesitant in terms of how the protein could be shaped, which throws a caution into the wind. It allows you to think about different types of designs you can modify so that you produce a high confidence that once translated in a physical cell becomes a real tangible functioning molecule. Yeah, and then you can also draw a lot of analogies, as I said, um, thinking about rigidity of molecules, flexibilities provided by these kind of loop-like structures. Um, and then you can also start to speculate and imagine. So one exercise that we did was to speculate a function and then you reverse engineer it in terms of molecules. So one of the people were thinking about creating a organic, sustainable car tire, which is not made of artificial rubber, synthetic rubber, um, but using engineered proteins that has really elastic property that can be used to inflate the vehicle, and then you can back engineer how the molecules could be shaped, and then back engineer even more, and then figuring out what sequences this particular protein would have. If you have that sequence, then you can also then put it into the lab and actually make the molecules using a biological colonies. Yeah, so these are the language of life with the DNA, which I mentioned, amino acids, which then translates into proteins. Essentially, these are matters that do things. They once amino chain is made, it falls into the origami shape like a 3D structure. This shape then dictates what the protein can do. So this is just the very basic fundamentals of biology that I teach to designers. The whole point is really to not. This is not really about learning biology. This is about operating at a very premature level, from a very high level in terms of molecules. What I really want to do, I mean, this is one of my personal goals, is to really push this idea that design can also be done from a molecular level. It's not just chemists or biophysicists to do this. They actually call themselves designers in their own right, which is a very credible claim, I would say. And what I want to do is I want to have this paradigm translated into conventional design communities as well. It might be a hurdle to navigate, but I think this is type of the level of thinking I think we should adopt, especially given the accessibilities of tools of biology and artificial intelligence. Yeah, and then I teach about how proteins could be design materials of its own right, as I said. Last week, it was really about cells and colonies of bacteria and mycelium and fungi. But now we are going a step back even further as thinking about proteins as design materials, which would bring about different types of challenges. Um, but at the same time, it can also provide real opportunities for designing pretty much without limitation. If you can design molecules, you can customize and fabricate molecules in a very, I would say, granular way, rather than dealing with macro structures like fibers or textiles. If you can really think on a molecular level, maybe there is more power to that, and you can be more specific and precise of what you can design. Yeah, and then we think about the alphabets of designing with life. We talk about the stickiness, flexibility, bulkiness, the charge of the molecule. So now it's really going down to the nitty-gritty of characterizing or even anthropomorphizing these molecules, which can be a little bit extreme, but this is actually not too different from chemistry lesson. So we identify some of the common amino acids that make up these protein structures. And then it's almost like learning the new alphabet of biodesign, learning what these characters stand for, how they're composed chemically, and why this chemical composition matters in terms of the final outcome. And we try to draw the personalities of these different characters into translating that into a physical property. So in nature, there are 20 different amino acids out there with different personalities, and we usually go into a lot of detail into how that works. Um, so proline, for example, is one of the more interesting amino acid to tinker with in this area. It has this kind of a loop-shaped structure, which allows the proteins to bend. So if you think about a really nice, safe, springy, coil-like texture of alpha helices in the protein, if you have lots of that, it's regarded as a very smooth and very flexible structure. But the way to engineer or the way to fabricate this material is to ask AI to produce more prolines in the structure, which then would translate into a more of a rigid and bendy, introducing these kind of sharp turns that are crucial for certain specific folds. So we are marrying the two worlds here, the idea of the chemistry of the different molecules and using the power of AI to actually suggest exactly which position within this sequence to modify. And this is why the acceleration of drug discovery and new molecule generation for commercial applications is happening at the moment. Tryptophan is similar to proline in a way that it has this kind of really large, bulky, and aromatic structure within its amino acid structure. And you can also, this is a really interesting one, again, from a designer's perspective. It actually allows you to stick different molecules on top of this amino acid. So an interesting example from a designer was that there was a designer I worked with. She was interested in engineering silk proteins, and the way she told me was her silk was traditionally to introduce color into the silk. They need to do some kind of mordanting process, I think it's called. It uses harsh chemicals to allow the silk to bind different colored pigments. So this process is very harmful, apparently. It's chemically very toxic, and it harms the environment because of these chemicals. So her idea was to use AlphaFold to find interruption points to introduce tryptophan into the silk protein. What this does is it allows the silk material to become more sticky, allowing the pigments to dye more easily. And this meant that she didn't have to go to this process of mordanting using harsh chemicals. And the idea was to create a sustainable silk that can be dyed and colored in a sustainable way because the engineered silk would be very color receptive and sticky to other pigments as well. So this is just one example of how it could be used. If you think about color generation and application in other biomaterials, including those in architecture, you can now start to think about how you can engineer proteins within a sustainable material to produce a certain color, but also certain textures as well. Yeah, lysine is also interesting one because it introduces a chemical charge, which allow molecules to either repel. So if you think about water resistance materials, if you're creating a jacket made out of bacterial cellulose, if you want to modify that to be more waterproof, you introduce lysine, which would repel a certain more water molecules to make it more waterproof. Next slide. Yeah, and there are 20 other examples. I don't want to go into too much detail. But yeah, essentially what we're trying to, I mean, what I'm trying to teach is that proteins equal shape, which then translate to physical behavior. So again, this idea of the mind shift of treating molecules as design materials and using AI to help within this process, I think is a very powerful combination. Yeah, we talked about silk. We talked about beta sheet structures, which is a slightly different structure to the alpha helices, which are more springy and soft. And then, yeah, we can talk about different types of applications. I just mentioned the silk earlier on, but it's a common material that's being used by companies like what's the modern synthesis, for example, and other companies that are using engineered living silk to create sustainable materials. They haven't really mentioned whether they are using AI to engineer or fabricate these materials, but I wouldn't be surprised with the accessibility and how good these models are becoming, that they would actually incorporate AI into their workflow in the future. Yeah, and hydrophobins are the proteins I wanted to mention in terms of waterproofing. So these proteins, they have a lot of lysine amino acids in their surfaces to repel water away from the material. And it's actually a fungal material, which is even more interesting because fungal materials like mycelium are prevalent in biodesign and is held as one of the future materials of bioarchitecture and biofashion. And I think, yeah, a lot of work will be done. I think the work is already being done on hydrophobics at the moment to really engineer and fabricate these materials for waterproofing of the material and beyond. Yeah, and color is one of the other examples of people are thinking about. So green fluorescent protein is, I would say, one of the goats, or if you one of the incredibly useful molecules that are used in biology and biodesign. It has a very specific fold. Unlike many other structures, it has this kind of barrel-like fold, which creates a pocket for a molecule called the chromophore to attach itself within this pocket. And this chromophore is what it actually gives the material its color. A common chromophore for GFP is the green chromophore, and it allows the material to glow under the UV light. And this has been used in biomedicine and other applications, almost like a LED light, as I mentioned last week, treating biocomponents as electrical components. And again, it has a certain shape, it has a certain pocket. If you can somehow engineer this pocket using AI, in theory, you can actually create a different types of chromophore pocket so that it produces a different color. So almost like engineering the LED light to emit different frequencies. This is exactly where the AI can actually intervene and engineer and introduce new colors into the spectrum for biodesigners. So just to sum it up, we have the ESM fold, AlphaFold 3, and RF diffusion. So these are some of the functionalities that we can actually do with the protein structure predictions, visualize confidence, export and review. And then, yeah, designers can use it, as I said, to think about form to following function and how these tools can be integrated within the workflows as well. Interpretation is a big thing in AI biology. There's a huge debate within the community about how trustworthy these tools are. And so it's actually one of my main research focus at the moment is to really critically evaluate these models and give some type of a measuring tool that you can actually use to indicate the trustworthiness of tools like AlphaFold. So a lot of research and um, yeah, data collection is being I'm doing at the moment is around this one, so that people like the designers who are interested in working with biomaterials and AI can gauge how trustworthy a certain models are. So this is the tool that I'm developing. And then, yeah, another way of thinking about it is to ideate and sketch using these tools to speculate and think about potential materials that you can create, and then back engineering what these proteins could be shaped and how it could be sequenced and manufactured. Another useful link that I always refer to is the AlphaFold server. You can also check it out in your own time. It has a huge databases of AlphaFold. So these are artificially generated structures that you can browse. And these many of them are not found in nature. So if you want to start a project and building proteins from these structures, you can go to them and analyze it for your own projects. And yeah, I would say this is one of the most useful tools out there at the moment. Next slide. Yeah, and then yeah, these are some technical details that I don't want to go too much detail into. Yeah, but there are different levels of confidence scores and analyzing the trustworthiness. If you're interested in these kind of things, you can do that. Um, yeah, and then you can do it in a more of a structured way like this. What I also uh, finished the talk and workshop is the potential consequences of using AI in biodesign.
Hmm.
A lot of people who take my workshops, they become very idealistic, and I have another set of slides. I'm not going to go through them here, but what I want to say here is that it's very promising if you look at so one of the designer talking about Silk I mentioned earlier, she was
Hmm.
very happy that she could go away and go to a laboratory and present her AlphaFold artifact and get them to design this protein for her and can manufacture it, which can then be used as a silk material. There is a lot of workflow processes that goes in, obviously, and people should not be too idealistic about the use of AI in biodesign in this way, because yes, you may be able to create a really high confidence score of the silk protein that would bind a lot of dye molecules. You may find a workaround that would be incredibly environmentally friendly and economically very viable as a product using these tools. But there are many hurdles and bottlenecks within this workflow. So AI to use molecular prediction is one thing, but the reality is that the physical components of lab
Hmm.
validation is a huge bottleneck in itself. And this is what I try to spell out and specify so that people can be less idealistic, but more practical and also responsible.
Hmm.
If you think about the validation of these explorations, they do cost time and money. Somebody has to do it.
Hmm.
Maybe in the future, robots can actually validate this for us, but nevertheless, it would cost energy, time, and money. And so this is something that you should be able to confide in other people and discuss and collaborate together to be more responsible on how AI could be how AI could be used to generate new materials and for new biodesign explorations.
All right. Thank you. Thank you
Yeah,
very much. I mean, thank you for this example with the silk. I think that make it like really, really practical for me, and,
yeah.
and I guess for most of the students as well. So how long did you, so you say you were working with that students? How long did that kind of take for both of you to figure out, oh, that might be the solution, or was
Yeah.
it like an obvious thing for you?
Yeah, I mean, it depends really, because
Yeah.
I mean, with the silk example, she already were working on
Ah,
the
okay.
silk for some time.
I see.
Um, but just to speculate and give a basic idea of what could be changed, I
I
think that
see.
can be done
Mm,
very quickly because
Okay.
Once you get the hang of the tools itself, you
Okay.
can start to incorporate that into your workflow. So you can actually give, let's say if you're working with mycelium, you
Yeah.
can go to the database and find the mycelium sequence,
Ah,
and then
I
you
see.
can
Okay.
ask,
That's how you do it. Okay.
the tools to actually suggest ways to make it more pliable or make it more rigid.
Ah, okay.
And then they would give you specific mutations you can introduce to your sequence. And
I
this is
see. I see.
what people are doing
Okay.
with things like a modern synthesis, the company I mentioned earlier. They're actually doing the very thing. They scan the
有一定要这样。
database, they find the functionality, and then they ask the tools to actually generate a new sequence for them.
Okay, okay. All right. That's and then so did she send then the her idea to a lab and then did the lab kind of accepted this idea and say, okay, well we can work on that or did
Yeah,
you give her like a price of okay, well that's gonna cost you, a million pound or I don't know.
yeah. I
Did she
haven't really
try
heard
that
back
or
from her, so I'm not sure whether
okay.
she got gone ahead. But
Yeah,
um, I suppose you can actually, yeah, propose that idea for sure.
yeah. Okay.
Some people are very skeptical about that, whether it's as straightforward as that. I
Yeah,
mean, certainly in terms of
I know.
ordering a sequence, it is very straightforward. It's not a mystery.
Okay.
You can actually get the molecule delivered to your house even.
Oh, really?
But
Oh,
whether
wow.
to
Okay.
inject that into a molecule, a cell to produce your protein, that's a whole different kettle of fish because you need a bioreactor to generate your proteins using a cell, and then you have to have a proper lab to actually, you know, house your bioreactor and purify that molecule. So
Yeah,
because
okay.
protein purification is a huge industry in itself. Because I
Oh,
was in
really?
protein purification
Oh, I see.
for 2 years, trying to purify
Uh,
a certain molecule, and it's not easy because it's a very messy mixture, right? Because if you have a bacteria
okay.
that produces your protein, your protein is mixed amongst zillions of other molecules, right? So
I
how do we
see.
fish that out? That's the big challenge.
Ah, okay. Okay, okay. So searching for that protein is still an issue actually. When you do
Yeah,
you said the protein cleaning, is that what you say?
exactly. Yeah, yeah.
Okay. Or is it still manual at this point?
Yeah,
Is
so
it kind of any
let's say if you
assist?
have
Yeah,
a silk that you want to
yeah.
extract from your mixture,
Yeah.
then yeah, you have to filter out all the other impurities and to make it scalable. So from I don't know, 100-liter tank of bioreactor, you probably get like a tiny, I don't know, 50 gram of silk, that's all it
Wow.
can produce. So you have to think about, be a little bit smarter in terms of how you can scale and what type of
Okay.
species you can use to really scale it up in the tangible level.
Okay. So it's all very well to kind of thinking about AI at the beginning,
Mm.
but that doesn't resolve the whole chain of work that
Yeah,
goes
although
afterwards. Yeah. Okay. Not
ChatGPT,
yet.
like GPT has been used in the lab workflows also.
Oh,
So they
okay.
would actually calculate based on the variable, what is the optimal condition for this bacteria to produce your protein. So it's going crazy at the moment. It's
Wow. My
incredible.
goodness. Great,
Mm.
great. Yeah. I hear a lot kind of also, I think a lot of, I wouldn't know, are they biologists or where somehow the use of AI has been somehow being pushed into their practice. And somehow
Mm.
they were complaining that they somehow become more managers where they were kind of
Mm.
more hands-on, but now they've just been kind of
Mm.
managers. Okay, all right, we're going to try this one, or we're going to prioritize this versus that, and some of them are
Mm.
somehow also, expressing their frustration because the
Mm.
job has kind of changed, and not
Yeah, yeah,
necessarily
absolutely.
in a good way, where there was a little bit of
Yeah.
creativity, there was maybe a little bit maybe of enjoyment. I mean, you were talking about kind of walking on the bench was probably a negative things, but
Mm.
might not be negative for everyone. There's
Yeah.
maybe I do not know. I have never worked on a bench. I have no idea. Do you have this
Yeah,
experience?
no, yeah, I agree. I mean, I think,
Mm,
I mean, who knows how this will change the way we work. I mean,
mm,
um, yeah, I mean, I think with the things like agents, I think they'll be a little bit more hands off now,
yeah.
especially in terms of lab work, that's for sure.
Mm,
But
mm,
whether or not these people will be made redundant, I don't think so, because there can be,
mm.
like, as you say, the managers of these workflows.
Yes, 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 yes.
Obviously the human element and the banter and the the social aspects would be gone because people would socialize in these laboratory
Yeah,
spaces.
no, I know. Yeah.
Mm.
And I don't know if
Yeah.
you uh what was that book? There's another book from Bruno Latour that was really interesting. Who
Mm.
was talking about the life in the laboratory. And
Mm.
the life of laboratory, it was him and Stephen Woolgar, who was a professor at I think at Oxford at the time, and they
Mm.
collaborated, and it was based on a kind of ethnographic observation of how a laboratory works. And I think you are
Yeah.
right. I mean, what you said, there's a lot of banter, and I think their big discovery was that, well, science is not produced in a scientific way.
Mhm.
Of course, there's scientific protocol, but there's a lot
Mhm.
of time spent in front of at that time in front of the fax machine. There's
Mm.
time, in front of the coffee machine, and there's
Yeah.
a lot of things that I kind of come across, like kind of human creation. That the stress,
Yeah.
like you probably had recently, of, oh, now we need to publish this paper, we need to go really really quickly, and there was
Mm.
this competition about who's going to be publishing this thing first. So
Mm.
this is the life of a lab, and I think he kind of describe. I think I think it's called The Life of the Laboratory. I think that's the name of the book.
Mm. Yeah.
It's still a really really good book. I really really like this kind of idea. It
Right.
expresses very very well, and even in architecture last year we used that book somehow to kind of say, well, if you want to create a lab in architecture, you're not just going to create the kind of all of the rooms of the lab.
Mhm.
Maybe you can
Yeah.
also kind of create the space where they will be meeting, where they be
Mhm.
where people will be forced to kind of to talk, to chat, and to kind of share their discovery within the same lab,
Mhm.
of course. Yeah.
Yeah.
So that's that to me, that really kind of rings and a kind of important element, that to be human there's a need of. I think you were talking about friction in terms of design. I really like
Mm.
the idea of friction in design, and I think to
Mhm.
kind of the design somehow is there to slow down things a little bit, to make us think, to
Mhm.
make us talk to one another, and not just being
Yeah.
part of a machine somehow. I think
Mhm.
that maybe the element of resistance that we need to kind of incorporate in that
Yes.
collaboration. I think
Yeah.
that's to kind of admit that we are not there. And the famous competition between human and machine. And we just need to admit that we are more now. We are more the ones that slow down. So maybe that's our power, is to slow down a
Yeah.
little bit. I don't know.
Yeah. Yeah. Indeed.
Um, we'll see. Any questions from the students? So three students say they were open to ask questions today, and the first one was Yunwen. Yunwen, are you there? God, I think it's going to be a bit like last week, but hopefully be better.
Yeah.
2 weeks ago was really great, I have to say. Really, really great, great
Wow.
moment. I don't know. Maybe they feel shy. It may be difficult
Yeah.
to ask any question with your topic. I
Yeah.
don't think so. I'm not a specialist
Mhm.
in this topic, so I think there's
Yeah. No
plenty of
problem. I
questions.
mean, feel free to email me also. I'm happy
Okay,
to
okay.
answer them.
So Yunwen, part of the assessment, you have to ask a question. It's part of the kind of English
Right.
element
Right.
of the talk. Yunwen, are you there? Again, I can ask once. Yunwen twice. All right. All right. Next one. Hui Xuan. Aha. Yes.
Oh
Someone
yeah.
say yes. Who is
Great.
saying yes? Can you open your microphone and identify yourself? That sounds very serious.
Yeah.
All right. Okay. I think it's time to find the microphone and turn on the microphone. Is that Yuan or is that Wei Xuan? Mm. Hello.
Hello,
Ah,
hello.
Oh, hello.
who is, oh, who is it? Who is it?
I'm you.
Yuan?
Yes, yes, yes. Can you hear me?
Yeah, no, I
Yeah.
can hear you now.
Ah,
Perfect.
yes.
Very good.
Thank
Very
you.
good. Please ask your question. Thank you so much.
Thank you for the great lecture. As an architecture student, I'm interested in how this idea can be applied to architectural design. Because you mentioned AI biology as data and responsive surface. And in architecture, when we use biology as a reference, We often stay in at the visual level, like copying just the shape of the butterfly. But from your lecture, I feel that
Mm,
biology design could go deeper than appearance. So
mm.
my question is, how can architects move beyond copying biology form and apply this idea in a more meaningful way?
Yeah,
Yeah,
that's a
that's good
great
things for me. Yeah.
question. Yeah, I suppose I think there's a room for interpreting. I mean, certainly from what I'm doing, which is on a molecular level of exploration, to find some type of visual language that resonates outside of conventions of biology. I don't know what that might be. I used some generative AI tools to visualize what these proteins could look like when it became a final structure, a final material. Um, I mean, I suppose that's one of the first kind of a low-hanging fruit that you could think about. Um, yeah, I think it's really understanding about the materials and yeah, the imaginations could be uh, one of the key things to use to generate these visualizations. But ultimately, it's very hard, I think. It's from a molecular level to how that would turn out to be the final material. Yeah, so I'm not sure whether I'm answering the question very well, but
Not yet.
developing your own way of visualizing, I think that's key. But in order for you to do that, you need to have a fundamental understanding of molecular properties and behavior.
I mean, the whole biomimetic kind of architecture. I mean, this is the one we were
Mhm.
talking about. The
Yeah.
form is, I just have to remind the student here, the form is just one of the element. Yeah. The idea of the biology as a system is a much deeper approach to the biomimetic architecture. Yeah, let's not forget that. I mean, there's been a few books on the topic, and I think from the one I've read, the one from England, for example, they mention really a whole series of level of engagement with the biologic. And
Mhm.
if you really kind of think the form is enough, I would say the form is probably the least interesting part
Yeah.
in my opinion. Whenever you kind of see a form, like you said, a butterfly. I mean, first of all, it's a very good question. I think your question is good,
Yeah.
but if we kind of focus on just the shape of things, we're really not understanding biology, in my opinion. We're really kind of understanding the kind of first level. And I think here Dr. Kim kind of pushes us to kind of go at a much lower level, talking about the kind of the relation we have with the bacteria, but also in terms of your building, how in terms of ecology, in terms of relation with the weather, in terms of which material might be the material of the future. I don't know. And in fourth years, as some students are kind of joining those special groups where they examine kind of new materials, and then those are, like you say about silk, yeah,
Mhm.
a material produced, a biologically produced material, there's still some imperfection in the way that we want to use them. So that's
Mhm.
where you can kind of apply your knowledge. But your knowledge has to be at the material level, at least, to kind of be to be able to kind of respond to what has been said in the last two talks.
Mhm.
You want to
Yeah.
add something? I'm trying to
Yeah, no, absolutely.
to
And it's
contribute.
funny because quite
Yeah,
recently, a lot of
yeah.
biomimetic designers
Yeah.
approached me because when I was
Ah,
talking
ah,
about these molecules,
yeah,
so
yeah, yeah.
there is a little bit of misconception as well.
Yes.
This misunderstanding that what I'm doing with these simulated molecules is it's a pure simulation, which is not
Yeah.
because we are dealing with an actual tangible
Mm.
molecules that can be
Oh, that's interesting.
manufactured.
Yeah. Mm.
Yeah. So there is that level of dissemination that I certainly need to do just to make sure that we are dealing with tangible molecules that are in real life.
Yeah, it's the combination of molecule that is new, but the
Mhm.
the combination of protein that is new. But those proteins do exist.
That's right.
Yeah,
Yeah,
that's the logic? Yeah, okay. Just make sure if I
exactly.
understand
Yeah.
properly. Biology was a topic that I studied in high school, so it was a long, long time ago. And this is way beyond what we were studying in high school. But
All
that might
right.
be the topic of future high schools, but that is not where I am, definitely.
Yeah.
Okay, hey, thank you so much for your question. That was a really good question.
Thank you.
Thank you for appearing. What about Hui Xuan? Are you there, Hui Xuan? Hui Xuan once, Hui Xuan twice. Okay, all right. So let's move to the last one. Okay, it makes me happy. At least there was one question. Nguyen Quang Huy. Yeah, hopefully you can correct my Vietnamese. The third student in the chat. At least I'm not challenging myself there. Quang Huy, are you there? Yes, no? Once, twice. All right. Okay. All right. Again, thank you so much. I
So
think you kind
are
of
they in trouble?
no, a little bit. Well, they lost 10 points in their assessment. Okay, so that's the reality of the assessment
Yeah.
process. But not you right now. Thank you again for her question. And I know it's brave to kind of open your microphone and then to throw yourself with a question. So I very much appreciate. But again, thank you very much. You kind of opened the world of possibilities there. And then I kind of a little bit. It's just in term, and I think it's very important. It's not just in term of there's AI, we can go faster. I think we talk about that, and I think that's really important. And also there's an element of ethic. So where do we find ourselves back in that loop? We quite often with AI we talk about we kind of try to reassure ourselves that we are part of a loop. So where are we in that loop, and then how we might be able to kind of intervene or correct or challenge a tool. That is kind of all kind of possibilities for us to remain in that loop.
Yeah,
I don't know if that's it's kind of a question that you are considering as well. But to me, that's really kind
absolutely.
of yeah, it's kind of really important. Of maybe even we talk about kind of considering science as something that need to go slower. Is that's where the relation and you work at La Roche, maybe where La Roche was maybe faster science, but when it's more for national, it's maybe more about kind of slow down a little bit. Uh,
Yeah.
Maybe reinterpreting the kind of social into the science. That's the kind of
Mhm.
Latourian question. There's
Yeah.
plenty of topics I think that should force us to kind of reread the classics a little bit of well, how does a lab work? What is actually
Absolutely. Yeah.
a good lab? And I think of sort of, I
Mhm.
don't know if you have a final word on this
Yeah.
topic,
Just
or maybe other topics that worry you at this point, or
yeah.
excite you. Quite, don't need to be always worried about things.
Yeah, I suppose one thing that I can close
Mm.
on is to look at maker spaces and open spaces that are available
Yes,
to
yes, that's true.
designers.
Yeah,
I think that could
I
be
like that. Yeah.
an interesting starting point because they don't have this agenda of efficiency. They don't have this notion of pleasing the stakeholders, you know. So without this kind of a limitation, you can be a little bit more open to explore whether it's molecules or bacterial cells. And I think
Mm,
these maker spaces do exist in Taiwan, and you can land yourself there and yeah,
mm,
explore it in that way. Because I think a lot of tools and
yeah.
facilities and equipments that there are available there. So
That's true.
yeah,
So, but one of the kind of citizen kind of approach, or the kind of citizen
yeah,
DIY citizenship. That's a term I quite like.
that's
DIY
right. Yeah, yeah.
citizenship,
I mean, this is the
or
community I
hacking.
belonged
Yeah.
to a few years
Oh,
ago, and I think this is the reason why Joel from LCC invited
yeah.
me for the workshop many
Oh,
years ago.
yeah, yeah,
You
yeah,
know,
yeah.
so I think that community still exists, and I think it's a good way
Okay.
to think about not just speed, but just to have some reflection of what you're actually doing, and like
Yeah,
you
yeah,
say, what the lab is supposed to be and
yeah.
what it could mean.
Okay, great. Well, thank you so much about this tour presentation. Okay, I
You're
hope everything
welcome.
goes well in Stockholm.
Yes, thank you so
If
much.
I
I will check out the museum. Yeah.
yeah, and then put your Viking hat once in a while. I am a Viking too. All right, I'll put it in your head anyway.
Yeah.
All right, thank you so much. Okay,
Yeah,
thank
thank you
you to
so
the
much.
students. Yeah, and thanks to the student who joined us today. Thank you for the question, Yuen, and have a great evening then. Yeah,
Yes,
or
and you
great
too.
afternoon.
Yes, thank you.
Thank you.
Bye-bye.
Cheers. Bye
Bye.
bye.